Hello and welcome to this short video from the Foundations in Medical Neuroscience course in which I'm going to introduce the basic biology of a neuron. My name is Professor Phil Newton, and what I hope you're able to do by the end of this short video is indeed to be able to describe the basic properties of a neuron. Here then is a schematic, a cartoon of a neuron taken from Wikimedia Commons. You can see some key features of it that we're going to talk through later in the video. The most important thing that I want to get across at this point is that a neuron fires an electrical signal that we know as an action potential. We'll talk about how that is generated in another video, but the key feature here is that the action potential travels in a single direction from what we might call the top to the bottom of the neuron. Detail of the structures then in the neuron: the top of the neuron are the dendrites, then there's the cell body or the soma, giving rise to the axon hillock, which is the top of this long thin structure called the axon, and then leaves the neuron at the form of the axon terminals. Lots of what we're going to talk about in Foundations in Medical Neuroscience involves the connections between neurons. So here we have two neurons connected together. So we have the axon terminals from one neuron and the dendrites for the next neuron. And the structure that connects them, or the gap between them, more correctly, is known as the synapse or the synapse. We're then able to talk about the presynaptic neuron and the postsynaptic neuron. In general, then, the action potential will travel down the presynaptic neuron. There'll then be a connection between the two neurons, and potentially an action potential will travel down the postsynaptic neuron. There are two key signals that neurons then generate as part of this process. The first, we've already briefly mentioned, is called the action potential. This is an electrical signal that travels within neurons, as indicated by the green arrow on the previous image. The signal that travels between neurons is known as a neurotransmitter, and this is a chemical signal. There are two basic types of neurotransmitter that are important for understanding the basic biology of a neuron. The first of these is an excitatory neurotransmitter. When the presynaptic neuron releases an excitatory neurotransmitter, for example glutamate, this increases the probability that the postsynaptic neuron will fire an action potential. So the presynaptic neuron releases glutamate or some other excitatory neurotransmitter that travels across the synapse, binds to the postsynaptic neuron, and that postsynaptic neuron then is more likely to fire its own electrical signal, the action potential. Inhibitory neurotransmitter has the opposite effect. This decreases the probability that the postsynaptic neuron will fire an action potential. An example of an inhibitory neurotransmitter is gamma-aminobutyric acid, otherwise known as GABA. We're going to go back briefly then to our cartoon of the neuron and talk about those yellow dots along the axon. These are what's called the myelin sheath. The myelin sheath that you can see here is formed of individual cells, specialist cells called Schwann cells. These form the myelin sheath of neurons in the peripheral nervous system. Cells in the central nervous system, i.e. in the brain, have a myelin sheath formed of specialist cells called oligodendrocytes. The myelin sheath is important because it is very simply electrical insulation tape for neurons. Some, but not all neurons have this myelin sheath, which means their electrical signal, the action potential, is insulated and it's able to travel more quickly with less leakage. This is important for a number of reasons, one of which is that when we talk in basic neuroanatomy about grey matter and white matter, the distinction between those is neurons that are myelinated and unmyelinated. Here is a coronal section through the brain, and you can see some of it looks white and some of it looks grey. Very basically then, the structures I should say, that are white because many of the axons of the neurons within those structures are myelinated. Myelin is a basic fatty substance, so it shows up white on these sorts of images. The neurons in the grey matter are in general not myelinated. The axons are generally shorter. This is important also because then the imaging techniques that we use throughout neurology and neuroscience often rely on the contrast between different structures, and lots of the contrast between myelinated and unmyelinated neurons shows up well in imaging systems because of the different chemical compositions, for example fatty and non-fatty, and the different amounts of water contained within. It's also important because there are certain diseases, for example multiple sclerosis, which are associated with a reduction or a loss in the myelin sheath, and that means that the affected neurons do not conduct their action potentials as effectively, with associated challenges for the basic function of the nervous system. We're used to thinking about presynaptic, postsynaptic, and the synapse. One final point I want to leave you with is that even though we will talk about this basic relationship a lot in Foundations of Medical Neuroscience, the nervous system, or the brain, is actually 86 billion neurons all connected together. Then there are a further 86 billion non-neuronal cell types, which we'll talk about in a different video. Lots of these are connected to multiple versions of each other, so one neuron will be connected to dozens, if not hundreds or thousands of other neurons. They'll all be firing action potentials in certain sequences in a coordinated way, which is what gives rise to the basic function of the nervous system. We can see this in action. Here is some imaging from Garrett Stuber at the University of North Carolina, in which neurons in a region of the brain called the hippocampus have been loaded up with a calcium-sensitive dye. When neurons fire an action potential, calcium is one of the ions that comes into the neuron. And what happens here, as you can see, is that when the neuron fires an action potential, calcium comes in and the neuron lights up bright. And you can see the firing of these individual neurons reflecting some sort of pattern as this awake, behaving mouse navigates its way around its environment. We can also record action potentials firing from individual neurons. This is a trace that does just that, and the blue lines across it represent individual action potentials. We can record this and then look for patterns in the firing of those action potentials within individual neurons and within groups of neurons, and you can actually hear, if you hook it up to an appropriate amplifier, the sounds of these action potentials. Here we go. Okay, that's it. Here are the credits for some of the images and the videos that we've just used. If you have any questions about the content of the video, please do send me an email, p.newton@swansea.ac.uk, and I'll see you in another video. Goodbye.